ICH Q1 through Q9 are the core quality Q guidelines that define how pharmaceutical products should be developed, tested, and controlled to ensure consistent quality, safety, and regulatory compliance. They cover stability, impurities, analytical validation, pharmaceutical development, risk management, and quality systems. Together, they form the scientific and regulatory foundation for modern pharmaceutical quality. Stability. Stability guidelines Q1A through Q1F define how a drug substance or product changes over time under temperature, humidity, and light. They specify storage conditions, testing intervals, and requirements for global submissions. ICH. They ensure manufacturers establish a scientifically justified shelf life and storage conditions. Main focus: stability of drug substances and products. What it regulates: storage conditions, testing intervals, stress studies. Key outputs and requirements: shelf life, retest period, storage conditions. Where it applies: development, registration, life cycle. Q2 describes how to validate analytical methods used for testing drug substances and products. It covers accuracy, precision, specificity, detection limits, quantitation limits, linearity, range, and robustness. These parameters ensure that analytical results are reliable and reproducible. Main focus: validation of analytical methods. What it regulates: accuracy, precision, specificity, LOD/LOQ, linearity, robustness. Key outputs/requirements: validated analytical procedures. Where it applies: QC labs, method transfer, regulatory submissions. Q3 impurities. The Q3 series sets limits and identification thresholds for drug substances Q3A, drug products Q3B, and residual solvents Q3C. It ensures that impurities are controlled to safe levels and that toxicological risks are assessed. Main focus: control of impurities. What it regulates: organic impurities, inorganic impurities, residual solvents. Key outputs/requirements: identification thresholds, qualification limits. Where it applies: API and finished product quality control.
Q4 Pharmacopeal harmonization. Q4 harmonizes general tests and monographs across major pharmacopoeias, USP, EP, JP. This reduces duplication and ensures consistent quality standards globally. Main focus: harmonizing pharmacopeal standards. What it regulates: general tests, monographs. Key outputs/requirements: unified global standards. Where it applies: QC testing, dossier alignment.
Q5 Biotech product quality. Q5 covers characterization, manufacturing, and control of biotech-derived products such as proteins, monoclonal antibodies, and vaccines. It includes guidance on expression systems, purification, viral safety, and comparability after manufacturing changes. Main focus: biotechnology-derived products. What it regulates: characterization, expression systems, viral safety. Key outputs/requirements: comparability protocols, biological specifications. Where it applies: biologics development and manufacturing. Q6 specifications. Q6A chemical drugs and Q6B biotech products define how to set specifications, tests, methods, and acceptance criteria for drug substances and products. They ensure that specifications reflect critical quality attributes and manufacturing variability. Main focus: setting specifications. What it regulates: critical quality attributes, acceptance criteria. Key outputs/requirements: finished product and API specifications. Where it applies, release testing, stability, regulatory filings. The ICH Q7 guideline provides international standards for good manufacturing practice, GMP, for active pharmaceutical ingredients, APIs, used in human medicines. It explains how APIs should be manufactured using controlled and validated processes, handled and stored to prevent contamination or mix-ups, tested and documented to ensure quality and traceability, packaged and labeled correctly and clearly, distributed under appropriate conditions. The guideline covers quality management systems, personnel responsibilities, equipment and facilities, documentation and record keeping, production and in-process controls, quality control testing, complaints and recalls. Purpose: to ensure that APIs are consistently produced and controlled to meet the required quality, safety, and purity standards worldwide. Main focus: GMP for active ingredients. What it regulates: personnel, equipment, documentation, production, QC. Key outputs and requirements. API GMP compliance system. Where it applies, API manufacturing sites. Q8, pharmaceutical development, QBD. Q8 introduces quality by design, QBD, encouraging manufacturers to understand product and process variables scientifically. It emphasizes design space, critical quality attributes, CQAs, and process understanding to ensure robust manufacturing. Main focus: quality by design. What it regulates: design space, CQAs, CPPs, process understanding. Key outputs and requirements: enhanced development reports, design space proposals. Where it applies. Formulation and process development. Summary of Q9 quality risk management. Q9 defines how pharmaceutical companies should make consistent, transparent, and risk-based decisions. It ensures that risks to patients, product quality, and regulatory compliance are systematically managed using scientific knowledge and process understanding. Main focus of QRM. Protecting the patient by ensuring product safety, efficacy, and quality, understanding processes to identify where risks originate, making decisions based on science and data, not assumptions, prioritizing resources toward high-risk areas, maintaining lifecycle control through continuous monitoring and review, ensuring transparency and documentation of all risk decisions. What QRM regulates or influences. Q9 does not regulate like a law, but it guides how risk thinking must be applied within GMP systems. It influences manufacturing processes and controls, process validation and ongoing process verification, equipment qualification and facility design, cleaning validation and cross-contamination control, supplier and material qualification, change control decisions, deviation investigations and CAPA, quality control testing and specifications, data integrity controls, packaging, storage, and distribution, product lifecycle management and continual improvement. Key outputs and requirements. QRM requires structured, documented, and traceable risk activities. Typical outputs include risk assessment, risk control, risk communication, risk review. Where QRM applies. QRM is used throughout the pharmaceutical lifecycle, including development, manufacturing, quality systems, distribution, and post-market, like defining CQAs and CPPs.